Hello, my name is John McLaughlin. I'm a research scientist here at Rutgers University, and today I'd like to talk about the use of functional genomics and how we use that in our lab to identify genes to a mycotoxin that is produced by an agriculturally important pathogen of wheat and barley called Fusarium graminearum. And so the title of the talk today is Unraveling Trichothecene Toxicity, a Cross-Kingdom Functional Genomics Approach. And what I'll show you is you can use a number of different, and we have a number of different model organisms to get at the function of a small molecule toxin like trichothecenes that I'll be talking about. So the outline for today's talk is just to go over, one of our goals in the Rutgers laboratory is to elucidate the mechanism of action of an agriculturally relevant small molecule mycotoxin called deoxynivalenol, also known as DON, and the common name is vomitoxin. And it's approximately 300 molecular weight small molecule. It's a virulence factor, and we're trying to understand how that functions in plants. And the most important thing that we're trying to relate this to is because it promotes infection, we'd like to understand the mechanism of that. And if we understand the mechanism, we can potentially design systems to prevent and protect. And the three examples I'll give today are using yeast, a green algae, Chlamydomonas, and Arabidopsis as model organisms to identify the resistance and sensitivity genes and the mechanisms of toxicity. So trichothecenes are toxic to eukaryotes. Yeast, algae, plants, mammalian cells are sensitive to these toxins, and I just give some examples here of, in this particular case, we have vero cells that were treated with a variety of different trichothecenes, T-Syn, DON, DOS, and T2. We showed that yeast are also sensitive. Chlamydomonas and Arabidopsis have different degrees of sensitivity. So what is functional genomics? I think you've spoken about this in your class, but I'll just tie it together with how we use functional genomics and how I define it. It's an extremely wide field that attempts to describe gene and protein functions and the potential interactions. And functional studies are aimed at discovering genetic variants and biological mechanisms. It's an active area used in plant improvement. One thing that's interesting is the merging of quantitative genetics and molecular biology, and using things like mapping causal SNPs to impact quantitative traits, like what we're going to talk about today, and some examples from a Rutgers laboratory. So the power of model organisms for high throughput screening and gene discovery. So in yeast, we can identify eukaryotic resistance and sensitivity mechanisms to trichothecenes if we screen a yeast genetic mutant library. With Chlamydomonas, we can do something similar, and this allows the mutant libraries of Chlamydomonas allows us to screen a photosynthetic algae mutant library to discover novel genes that play a role in trichothecene toxicity. And then Arabidopsis, I'll show an example of a genome-wide screen using activation tagging to identify gain of function mutants that provide protection against the toxin. So this is an example of using a mutational approach to identify genes that, when overexpressed, give resistance. So some of the advantages of yeast. Yeast have rapid growth. They exist in diploid and haploid forms that allow mating, so you can mate in plasmids and mutations. That's especially important when you're doing like double or triple mutants on a large scale. It has a versatile DNA transformation system. The availability of yeast knockout strains that covers a large percent of the yeast genome is available. There's also overexpression libraries. In addition, there's the essential gene library, which instead of having the gene knocked out, they have a temperature sensitive version of the allele. And when you raise the temperature, that protein falls apart. And also there's this idea of synthetic genetic arrays for discovering genetic networks. We won't focus on that today, but that's a very powerful advantage of yeast genetics. And of course, the yeast, the Saccharomyces genome database is a model for a model organism for literature, for gene databases, and for just a wide variety of tools for using something like taking advantage of the yeast as a model organism. And I encourage you to visit that. So, for example, in our particular case, we had access to the yeast deletion library, and that's made up of around 70 96-well plates that are arrayed of individual and identified yeast mutants. There's approximately 5,200 or so. And if you array those on 96-well plates, in some cases they're duplicated and there's some blanks. So overall, that equates to about 70 96-well plates. What you can do is you can do something like this. You can put your toxin, in our case is a small molecule, in the media of the yeast media. And because yeast are sensitive to trichotesins, you can screen for yeast that are more or less sensitive. And in this particular case, we screened on the left-hand side is 4 micromolar tecin. And then on the right side is a doubling of that concentration, 8 micromolar tecin. So you can see on the 4 micromolar tecin plate, most of the strains are not growing very well. And there's some that are highlighted in the white and black circles that are showing more vigor than others. And this would be an indication that these knockouts play a role in sensitivity. And when you knock them out, they are more resistant. And then when you double that concentration, almost everything fails to grow except those 2 mutants. So one great thing about using yeast is you can screen in a high throughput manner, and you can use various, you can screen on different times, different concentrations, and allows you to identify, the extent of resistance of different mutations on a genome-wide scale. And when we did this, we identified some mitochondrial mutants that were of interest. And when we found that, we could ask specific questions. And this is just one example of that. So we found that trichothecines impact the mitochondria. And when we learned that, we obtained a plasmid that allows you to direct GFP to the mitochondrial membrane and look to see what the mitochondrial morphology looks like under different stresses. And in this particular case, the stress is being a trichothecene exposure. And when we looked at the control, we see this nice spaghetti network that is typical for yeast. And when you apply trichothecenes, you get a fragmentation of that mitochondrial network. And so that just gives us some clue about how the toxin's functioning. And it gives us some hypotheses about how this toxin affects eukaryotic organisms. And I'll show you that in the next slide. So once we identified that mitochondria is a target of these toxins, you can ask questions like, do trichothecenes inhibit mitochondrial translation? We know trichothecenes, one mechanism of toxicity is inhibiting cytosol translation, but we wanted to see, because the genome-wide screen directed us to the mitochondria, do these toxins also impact mitochondrial translation? So if we look at the far left, this graph is percentage protein translation relative to toxin control. And on the far left, we did an in vivo assay where we fed the cells S35, and we were able to measure the mitochondrial protein production using S35. And if you're looking at the total protein translation with 1 micromolar tecin, you can see is actually boosted. It's above 100% because sometimes these toxins have a toxin impact where actually the production of proteins can be induced. And that's an example of hormesis. And at that same level of trichothecene, the mitochondrial protein translation is seen to decrease relative to 100%. Now, of course, when you increase that from 1 to 4, that total mitochondrial protein translation goes down. And then if we look at in more detail an in organello assay, we also see that trichothecene having an impact on translation. So this is a case where you take the mitochondria, you separate the mitochondria from the cell, and you do the assay only on the mitochondria, and that's an in organello assay. Further proof that these toxins impact mitochondria directly. And so there's a lot of great tools that would enhance the speed of doing these types of assays. So these mutants are barcoded, and so each individual mutant can be distinguished from one another by that barcode, and that allows you to do some interesting things like pool these yeast mutants and then expose them to the plus drug or minus drug situations. And then by analyzing the DNA of the culture after a certain period of time, you can really identify how well particular strains do in the presence of minus drug and plus drug. And that amount of DNA of that barcode will give you an indication of fitness, and you can do this on the genome-wide scale, and it gives you a very rapid method of looking at fitness in the presence of drugs or other conditions. You could use a drug or heat or other conditions like that. And there's also techniques of using high throughput screening using robotics. So here's an example of the Singer Rotor had robot for screening yeast. So using this type of robot, you can screen, 6,000 yeast mutants on various plates and other conditions very rapidly. I would say within 3 hours, you could screen an entire yeast mutant library on several different concentrations of toxin. And you can do it at high density. You can do this at 1,536 density or even higher. So you can replicate that 96-well plate on the same plate, or you can consolidate 4 plates into one, for example, and have that at 384 density, and then replicate that 384 density quadruple times on a single plate, if that type of screen works for you. And again, we're relying on fitness or the growth of the size of the colony. What's interesting, there's computer programs that will go in and automatically measure the size of the colony and give you fitness scores. So you can do this relatively quickly. And the other thing I'll mention is the essential screens. The essential genes can also be screened using the particular libraries, that, for example, the temperature-sensitive mutants. And in this particular case, we've just printed the plates, looked at them at 25 degrees, and then shifted the temperature to 34 degrees, where that temperature-sensitive protein is no longer functional. And we can see differences between the sensitive and resistant mutants using this approach. The other interesting thing, valuable library that they have, is the overexpression library. So there's a library that overexpresses each of the 6,000 genes. And again, it's in an array, and you can identify unique mutants that give you resistance. And in this particular case, we screened the overexpression library, and we identified a microprotein that when overexpressed gives high resistance to trichothecenes.